Hi, we're going to go ahead and finish up our notes on evolution now. This time we're going to be talking about the evidence of evolution that Darwin presented and a little bit of evidence that we now know about since we know more about DNA and genes, things like that. So I just want to start back up with where we left off last time. We talked about natural selection, and natural selection, again, is remember that when the organisms that have the best traits for a particular environment live to reproduce and produce offspring with the same traits. So remember, there's two parts to that. One is that they're living long enough, and the other one is that they then reproduce. So both parts are critical, living and reproducing. Okay, and remember also that it's populations evolving and not individuals. Okay, an individual person or an individual flower does not evolve. It's the population together as a whole. All right, so we're going to talk about lots of different types of evidence. We're going to start with the fossil record. The fossil record compares fossils of older layers of rock with fossils from younger layers. So if you have a rock layer, what you should know is that the youngest are at the top and the oldest are at the bottom. They do know that age changes because of distinct coloration in the rock and other marks that they can find. So there's different layers that you can see being laid down. The youngest is at the top, the oldest is at the bottom. And that has showed that life on Earth has definitely changed over time. There's definitely changes from the fossils that were here a long time ago compared to the organisms that are living today. Remember, that was one of Darwin's initial observations that he made when he was on his trip on the Galapagos Islands. The next thing is the geographic distribution of living species. So what that means is that if you compare organisms that live on different continents, they are similar to each other, but they really don't look alike. They don't act exactly the same way anymore. And so what that is, the conclusion that you can draw from that is that they had different ancestors, but they have similar environmental pressures. And so that's called convergent evolution. So they're living in the same place, so they've evolved similarly, but they're still different. And so you can see some examples of that. In North America, we have like the beaver and the muskrat. And in South America, you have the capybara and the coypu, which are so the beaver and the capybara are similar to each other. You can see they look very similar, but they are different species. They don't have the same ancestor. But since they live in a similar climate, they had similar pressures put on them, similar competition for resources, so they have similar features. Okay, the next type of structure is called a homologous structure, or the next type of evidence, sorry, is called a homologous structure. So the prefix homo means the same. Okay, it's important to make sure you know what that prefix means. We'll talk about it a lot. So homo means same. So homologous structures are body parts that have the same structure but a different function. Remember when we did our height lab last week, we talked about how we have one long bone and then two bones in your forearms and then wrist bones and hand bones and finger bones. Almost every vertebrate animal has that same setup. So crocodiles have that same setup, whales have the same setup, birds have the same setup. So it starts off with one long bone, two bones here. For us, it's called the radius and the ulna. You then have your wrist, your hands, and your fingers. So you have the same structure but a different function. So bats use theirs for flying. We use ours very differently from that. And then you have other things that use it for walking. Again, very different from us. But what that shows you is that we had a common ancestor because we have the same structure. But now since we live in different places and do different things, it's a different function. Okay? The next thing is called a vestigial organ. A vestigial organ is a structure that has been greatly reduced and with no apparent function. So that means that, again, indicating a common ancestor. Make sure you have jotted down that vestigial organ is a common ancestor. An example would be your appendix or your tonsils, things that have no real function, but they used to. So in some organisms, an appendix is still used. So that's evidence for evolution, because why would we have stuff that we don't need? Our tailbone is also another example of that. All right, then the third type of structure is called an analogous structure. And an analogous structure is a body part with a different structure but a similar function. So a different structure but a similar function. So this is no common ancestor. And it's because of similar environmental pressures. So we have lungs, insects have trachea, and so that would be an example. We have the same function to get oxygen from the air instead of the water, but our structure is very different. Okay? So that's some evidence. Here's an example of the homologous structures. Homologous structures. That is what is an example of. So everything has this long bone called the humerus. And then you have two bones here, the radius and the ulna. You can see that some are thicker than others, some are longer than others. And then you have wrist, hands, fingers. Wrist, hands, fingers, the same thing here. Excuse me. You can see though that they are different structures. They have the same basic structure, but now some are thinner, some are thicker, but they have different functions. So our common ancestor would be some sort of fish-like structure that started off with that structure. Okay, so now up to some new evidence that's being used since we've done some more research with DNA. So Darwin would not have used this evidence, but we do know about it now. So what we know is that the reason that we have natural variation in our populations is because of DNA, and that's carried on our genes and our cells. And then the differences in DNA is what gives us variation. So if we have similar genes, then we have a common ancestor. And you can pretty much just go ahead and count the differences in nucleotide sequences, or you can count the differences in amino acids. So nucleotide sequences is for DNA, and amino acids is for protein. And so you're just comparing compounds to see what's similar and what's different. When we talk about genes and evolution, what we talk about is the gene pool. And the gene pool are all of the genes that are present in a population. All of the genes, all of the different alleles that a population has. And so that we can compare them from one species to the next. Okay, the last thing that we need to talk about then is what happens during this selection. So if you start off with the dashed line, so in each case the dashed line is your original population, and then the solid line is the new population. The first type of selection is called directional selection. So directional selection is when the curve stays about the same size and the same shape, but it shifts directions. So at one point in time, if you look at beak size, and here in the middle is what your beak size was favored for, a pretty average size beak. But then over time, in this particular population, the end was favored for. So a larger beak size was favored for. So the prevalence of birds with this larger beak size became more prevalent. Okay, so let's write it down. Occurs when individuals at one end of the curve have a higher fitness than individuals in the middle or at the other end. So it's the same shape curve, and it pretty much looks the same, just shifted. So you're now selecting for a larger beak size. So when we say selecting for, that means that those organisms will live long enough to reproduce. I would also like you to draw the graphs in your notes so that you have a visual representation of what it looks like. The second type is called stabilizing selection. So stabilizing selection, you can kind of see, starts off with the same type of bell curve, but now your curve gets really high in the middle and not very high on your ends at all. Okay, so that's called stabilizing selection. And that's when individuals near the center of the curve have a higher fitness than either of the others. And that example is with human birth mass. It's pretty much stabilized as to being between 7 and 8 pounds is where most babies are born. So not very many extremes to that. You don't have very many 12, 13-pound babies and very many 2 or 3-pound babies that will survive. Okay, the third type of selection is called disruptive selection. And you can see this is where the one in the middle is being selected against, and those at each end are being favored for. So now you end up with 2 peaks instead of 1 peak. The ones in the middle do not have a very high fitness. So that means that these in the middle would die and not be able to reproduce, whereas the ones on each end are both able to survive long enough to reproduce and pass their genes on. Okay, that's it for the types of evidence of evolution. We'll work some more with this in class. I would like you to go back into Moodle and fill in the questions that are on your Google form. Thanks.